The parasite to be discussed in this lecture is Echinococcus granulosus, which is also called hydrated for the dog tapeworm. Like other tapeworms, this parasite is also a ribbon-like tapeworm having flat body. Suckers are present on its calyx or head, and hooks are present on the rostrum, and there is a short strobila. The length of this parasite is just 3 millimeters to 6 millimeters and has three segments or proglottids. And among these, there is an immature proglottid, a mature proglottid, and a gravid proglottid. The picture of the parasite, the head portion having four suckers and hooks on the rostrum, a small neck portion, and then there are three segments of the strobila. The first one having terminal mass, the immature proglottid, then there is a mature in which there are complete sets of male and female sex organs are there, and the last one is the gravid proglottid, which is full of eggs. About the life cycle of this parasite, it is a zoonotic disease, and the definitive host is dog and other puneri. The parasite, starting from this stage, parasite lives in the intestine of the dog, attached with the help of hooks and suckers to the mucosa of the intestine. When gravid proglottid ruptures, gets separated, moves towards anus, it is excreted in the feces, and the eggs are released on the vegetation in the environment. The oncospheres hatches, and these oncospheres, they are ingested with vegetation, present on the vegetation by the intermediate host, which can be sheep, goat, cattle, or poultry. The oncospheres hatch from these eggs in the intestine of these intermediate hosts, that is sheep and goats. These oncospheres, they penetrate the intestinal mucosa, get entry to the circulation, and they reach different organs of the intermediate host, especially the liver and lungs. And in the liver and lungs, the cysts or large cysts are formed, which are shown here with a thick connective tissue wall, and in it there is large amount of fluid and root capsules and daughter cysts. These cysts, called hydrated cysts, they will be present in the liver and lungs and other organs of the intermediate host, sheep, goats, and cattle. Now, the dog becomes infected by eating these organs of the intermediate host, sheep, and goat. Now, the liver and lungs, harboring these hydrated cysts, they are eaten by the dog, and in the intestine of dog, protoscolocysts are released from these cysts. These protoscolocysts, they develop to scolocysts, which get attached to the intestinal mucosa of the dog. In this way, the life cycle of the parasite goes through the dog, the definitive host, and the sheep and cattle, that is the intermediate host. Revising life cycle once again, the adult worm of Echinococcus granulosus, which is about 3 to 6 millimeters long, resides in the small bowel of the definitive host, which is the dog. The gravid proglottid release eggs that are passed in the feces of dog, which contaminate vegetation. After ingestion of contaminated vegetation by a suitable intermediate host, it may be sheep, goat, cattle, or poultry, the egg hatches in the small bowel and releases an oncosphere. This oncosphere, that is the very parasite, penetrates the intestine of sheep, goat, etc., and migrates through circulatory system into various organs in these animals, especially the liver and lungs. In these organs, the oncosphere develops into a cyst that enlarges gradually, producing protoscolosis and data cysts that fill the interior. And this is a picture of the hydrated cyst, which contains a large amount of fluid in it, root capsules, and daughter cells. The definitive host dog becomes infected by ingesting the cyst containing organs of the intermediate host. After ingestion by the dog, protoscolosis evaginate, attach to the intestinal mucosa, and develop into adult stages in 30 to 80 days. Now the question arises how the human being becomes infected. The humans become infected by ingesting eggs, contaminated vegetables, fruits, etc. This happens incidentally with resulting release of oncospheres in the intestine and the development of cysts in various organs of man, especially the liver, lungs, brain, etc., etc. Well, keeping in view the life cycle of the parasite, we can think about pathology. The adult stages present in the dog, they cause no disease in the dogs, but significant changes occur in humans. In humans, slowly growing cysts put pressure on surrounding tissues and produce chronic space-occupying lesions. That is, chronic space-occupying lesions will be formed in involved organs, especially the liver, lung, brain, and other structures. There may be organ enlargement accompanied by a variety of clinical signs depending on size and location of the cyst. Compression of liver may result in jaundice, portal hypertension, and abdominal distension. Cysts, if they are present in the lungs, there may be hemoptysis, dyspnea, and chest pain. Cysts, if present in the brain or spinal cord, can provoke acute responses and numerous neurological sequelae, including epilepsy and blindness. Cyst rupture can occur sometimes, and it is associated with acute clinical signs such as peritonitis and pneumothorax, and the sudden release of hydrated fluid, severe allergic reactions such as asthma and anaphylactic shock. Protoscolosis released from ruptured cysts can form new hydrated cysts in the tissues they come in contact with. So in this case, and this can occur even during surgery when the cysts can rupture accidentally, and the protoscolosis, they come out and they can form many cysts in the tissues that they come in contact with. The hydrated cyst, well, as multiple organs may be involved in the clinical information, X-rays, and ultrasound findings, they all help in diagnosis. Serodiagnosis is possible, in which crude hydrated cyst fluid is generally employed. At present, the best available diagnosis is obtained by using combinations of all these tests. Thank you.